As we have seen that how light powers the process, but now let's look at the actual building block, that is carbon dioxide. Carbon dioxide is the source of carbon for synthesizing glucose. In nature, this is actually the most common limiting factor, because even though carbon dioxide feels like a lot to us, it only makes up about 0.04% of our atmosphere. That is a very tiny amount for a plant to work with. Just like light intensity, if you increase the concentration of carbon dioxide, the rate of photosynthesis will rise, but eventually it will plateau, means it will be stopped here, it will make a plateau here. So at that point the assembly line is moving as fast it can, and adding more carbon dioxide would not help, because now the plant is likely waiting for more light or a higher temperature for to help the rate of photosynthesis. So think of it as a bottleneck. The light energy will be kept constant, then definitely the carbon dioxide will only affect the rate of photosynthesis. So as we have seen how light distance affects a plant, but what happens if we keep the light steady and change the food supply? In this experiment, we are investigating how the concentration of carbon dioxide impacts the rate of photosynthesis. In this case, our independent variable is the concentration of carbon dioxide. We shall control the concentration of carbon dioxide by using different percentages of sodium hydrogen carbonate. For example, comparing a 1% solution to a 5% solution, then in that case we can increase or decrease the concentration of carbon dioxide in the solution. What are our dependent variables? Dependent variables remains the same. We are counting the number of oxygen bubbles produced per minute. This gives us a clear measurable speedometer for the rate of photosynthesis. And because we are only testing carbon dioxide, we have to be very strict with our control variables to ensure a fair test. Here, the control variable will be the light intensity. Okay, here the lamp will stay at a fixed position. Okay. In the previous experiment where we were measuring the effect of light intensity, we are moving this light lamp away or further, further towards the experimental setup. But here we shall keep it on a single place, on one place. Okay, we shall not move it. If we move, because if we moved it, we would not know if the bubbles are being produced under the effect of light or under the effect of carbon dioxide. That is the reason we shall keep light intensity constant. And second constant variable is temperature. We shall use a thermometer to monitor the water bath. And remember, photosynthesis is an enzyme controlled reaction. Okay, so even a small rise in temperature from the light, it could cheat the results. And here we are using the same plant, okay, same species and the same size of pondweed as we used in the previous experiment. So by keeping these factors constant, we can prove that any increase in bubbles is purely because the plant has more carbon building blocks to work with. Now let's look at the engine of the experiment. We place our aquatic plant inside a solution of hydrogen carbonate. This is not just water. Okay, I'm talking about this water. Okay, this is not just water. It is a controlled environment that provides a steady supply of dissolved carbon dioxide. So as the plant photosynthesizes, it produces oxygen gas. Because the plant is underwater, the gas forms bubbles. You can see these here. It forms bubbles. So we use an inverted funnel to catch these bubbles and funnel them up with an inverted test tube. This allows us to clearly see and count the gas being produced. One of the most important parts is the large water filled cylinder, this one. Okay, large water filled cylinder. This acts as a water bath. Lamps can get very hot and heat would speed up the enzyme reaction in the plant, giving us a fake result. Okay, uh, the water bath absorbs that heat, ensuring that only the variable which we are testing is carbon dioxide. So we shall keep, this is very important. part of this experimental setup. So this process is very simple. Switch on the light and wait a few minutes. This wait is very crucial. You must let the plant equilibrate or adjust to the new conditions. Then you count the bubbles for exactly 1 minute. What will be the result? As you add more sodium hydrogen carbonate in this solution, the rate of photosynthesis climbs up. It will increase. But just like with light, it eventually plateaus. You can see that in this graph. So at that point, the plant has all the carbon dioxide which it can handle, and it is likely waiting for more light or a higher temperature to go any faster. As we have looked at the energy, that is light, and the building blocks, that is carbon dioxide, but now let's look at the workers in the factory, which are enzymes. Photosynthesis is a chemical process which relies heavily on these biological catalysts, which we call enzyme. So the dark reaction where we combine hydrogen and carbon dioxide to make glucose is purely enzyme controlled. Temperature is all about kinetic energy at low temperatures. Okay, so at low temperatures, the molecules move slowly. There are fewer collision between enzymes and their reactants, so the rate is very low. But when we reach at an optimum temperature, this is the Goldilocks zone. The enzymes have maximum kinetic energy and work at peak efficiency. The rate of photosynthesis is at its highest rate. But what will happen if we further increase the temperature? This is the danger zone. If the temperature is more increased, it will get too hot, usually above 40 degrees Celsius or above 45 degrees Celsius. The enzymes become denatured. Their active site changes shape, they lose their function, and the rate of photosynthesis drops sharply to zero. This we have the if impact of the effect of temperature on enzymatic reactions that we have completely covered in detail under the chapter of enzymes. Okay, but here is also a good recap of that change in temperature, of change in enzymatic reaction under the effect of temperature, that with the increase in temperature more than the optimum level, the enzymes get denatured, and definitely the enzymatic reaction will get stopped. Interestingly, okay, a rise of just 10 degrees Celsius can often double enzymatic activity until the optimum level is reached. This does not help the leaves make food, it also helps the root actively transport minerals from the soil. Now, for your exam pro tip, you should pay very close attention to the shape of these graphs. Okay, these three graphs, pay a close attention to these graphs. Graph are light and carbon dioxide will always show a plateau. So if an exam asks why it plateaued at point X, don't just say it reached its maximum. Identify the bottlenecks. You can say that light is no longer the limiting factor. It is not limited by carbon dioxide or temperature. But what about the temperature graph? That is different. It is a bell-shaped curve. You can see this is a bell-shaped curve. Okay, if you see the rate crashing down after a peak, you know the enzymes have been denatured. As we have seen how raw materials like light, intensity, and carbon dioxide affect a plant, but now we are going to test the engine itself. Photosynthesis is controlled by enzymes, as we just studied in previous slide. Okay, photosynthesis is controlled by enzymes, and enzymes are highly sensitive to heat. So in this setup, when we are going to investigate the effect of temperature, we shall keep temperature as an independent variable. We shall use a hot plate or a controlled water bath. You can see this is a hot plate. Okay, we shall use this hot plate or controlled water bath to systematically raise the heat for each trial. And what will be our dependent variables? The dependent variables is still the rate of photosynthesis, because we are going to investigate the effect of temperature on the rate of photosynthesis. So dependent variables is rate of photosynthesis, which means which we shall measure similarly by counting the oxygen bubbles being produced per minute. And to make this experiment as a fair test, we must be incredibly strict. The light intensity must stay exactly at the same point, so the lamp does not move. The carbon dioxide concentration must stay constant using our sodium hydrogen carbonate solution at a same concentration. Because if we change any of these, we would not know if the temperature is truly the cause of the change in the bubble rate or there is something else. Now the thermometer is the most important tool here. Okay, we would not just trust the hot plate setting. We shall measure the water temperature directly. So my dear students, as the temperature rises, you will see the bubble count increases as the molecules move faster and collide more often. But be careful. If you go too far past the optimum temperature, the bubble rate will suddenly crash. This is not because the plant is tired. It is because the heat has denatured the enzymes, permanently changing their shape, so they can no longer do their job. Experiment beautifully demonstrates the Goldilocks nature of our biological system. Now that we understand the theory, let's walk through the actual steps you would take in the lab to collect your data. First of all, we set up our apparatus with the lamp at a fixed distance. And remember, we only want to test temperature, so the light must never move. We use a hot plate to reach our first target, say likely 10 degrees Celsius, which is very crucial, okay? And you must allow time for the plant to adjust to the new temperature before you start counting. It may take a few minutes, it may take a few moments, but you will have to wait for getting good results. Then we count the bubbles for a set time, usually 3 minutes, and record it. But we do not stop there. To ensure our results are reliable, we repeat the entire process at least 2 more times for every temperature. This allows us to calculate an average and spot any anomalies. Okay, then we then repeat the same process for 20 degrees Celsius, 30 degrees Celsius, and for 40 degrees Celsius. What should we expect to see here? From 10 degrees Celsius up to about 30 degrees Celsius, the rate of bubble production will climb. As the temperature rises, the enzymes and molecules will gain more kinetic energy, moving faster and colliding more often. But watch what happens after the peak. At very high temperatures, usually above 40 degrees Celsius, the rate crashes. The heat is now too intense, causing the enzymes to denature. They lose their shape, they become inactive, and definitely the photosynthesis process stops entirely. So whenever you plot this graph, you would not get a flat plateau like we did with light or we had with carbon dioxide. Here you will get a distinct bell-shaped curve, which you can see in this video. Okay, you will find a bell-shaped curve with a clear peak at the plant's optimum temperature. In that case, you can find out an optimum temperature for a particular plant species. Up till now, we have looked at the big three: light, carbon dioxide, and temperature. But in the world where we live, other external factors can act as a handbrake on photosynthesis. For example, first of all, let's talk about water scarcity, water shortage. Okay, water shortage leads to what we call the stomatal paradox. So when a plant is thirsty, it closes its stomata to prevent water loss. It is trying to save itself from drying out, but in doing so, advertently blocks carbon dioxide from entering the leaf. So a lack of water indirectly makes carbon dioxide as a limiting factor. Then we have environmental poisons, that is herbicides like paraquat, which are designed to kill weeds by blocking the conversion of light energy into chemical energy, literally turning off the plant's power supply. Similarly, sulfur dioxide, that is another poison, which is a major component of acid rain, that is a silent killer. Okay, it physically destroys the palisade mesophyll cells. Sulfur dioxide kills palisade mesophyll cells. Since these cells, which are palisade mesophyll cells, are the primary food factories of the leaf, so if we destroy them, it means the plant can no longer produce glucose regardless of how much light is available. And here is your exam pro tip. Do not forget the importance of minerals, specifically magnesium. Okay, we shall definitely get a greater look uh, when we shall discuss the role of essential minerals in plant growth and productivity. So here is just a very little note on it, that magnesium is the central atom of the chlorophyll molecule. Think of it like the engine in a car. No magnesium means no chlorophyll, which means the plant cannot capture a single photon of light, and this leads to chlorosis, where the leaves turn yellow and the plant eventually starves. Uh, this is a recap of to understand the logic of the stomatal paradox. Okay, stomata are the biological gates of the leaf. We have already discussed the guard cells and the stomatal closure and opening in the previous lecture. Okay, this is just a recap of that. The stomata are the biological gates. Those are gatekeepers of the leaf. They face a constant dilemma. If the plant is dehydrated, it must close its stomata to prevent further water loss through the process of transpiration. So if the stomata are not closed, the cells will lose their rigidity, then the plant will wilt and it may die. Secondly, by closing these gates is to save water, the plant physically blocks carbon dioxide from entering the leaf, because the gate to enter carbon dioxide in the leaf is the stomata. And if those are closed, it means the entry of carbon dioxide is also blocked. Even if the sun is shining brightly and the temperature is perfect, the rate of photosynthesis will plummet because there is no carbon dioxide. In this specific scenario, water makes carbon dioxide as a limiting factor. So here is a critical concept that often separates grade B students from grade A students. Okay, that is the instrumental paradox. For example, if there is a hot, dry afternoon, then the plant is losing water faster than it can absorb it from the soil. To save itself from drying out, the plant triggers its guard cells to close the stomata, which is a survival move, and but it comes with a massive cost. What is the cost? The entry of carbon dioxide is also gets closed. So, my dear students, with this stromatal paradox, we end our today lecture. I'm very hopeful that you have completely understood the limiting factors which affect photosynthetic process. In next video, we shall discuss about photosynthesis in relation to the environment and how these factors are being controlled in greenhouses to maximize the crop productivity. And we shall also discuss about essential minerals which the plants require for their better growth. So my dear students, see you in next video tomorrow with new lecture. Till then, goodbye.